Hello, everyone. Thank you for joining us for our November Research Spotlight webinar. I'm Dr. Sarah Hernandez, the Director of Research Programs for the Hereditary Disease Foundation. I want to start by thanking our sponsor for this webinar, which is Neurocrine Biosciences. And I also want to thank everyone who supports the HDF. It's because of you that we're able to fund exciting science that's transforming what we know about HD. And a big thanks to all of you for tuning in. Thank you so much for being here. I'm happy to be able to introduce everyone to 3 HDF-supported young investigators. Young investigators really are the future of Huntington's disease research. They are the ones that are learning to organize and implement the research projects that are shaping ongoing research for HD. They are also the future leaders of the top HD labs around the world. Today, you're going to hear from 3 of these people who will each share their work for which they received HDF funding. We're joined today by Dr. Katherine Croce, Dr. Joe Hamilton, and Dr. Chiara Scaramuzzino. Katherine is a postdoctoral research fellow in the lab of Dr. Yamamoto at Columbia University. The main focus of her research investigates how protein clumping and buildup contributes to neurodegenerative diseases like Huntington's. She's currently working on a potential therapeutic approach to combat this issue with the goal of slowing disease. Joe is a postdoctoral fellow in the lab of Dr. Sarah Tabrizi, who is looking at DNA repair proteins as modifiers of Huntington's disease. And he has a primary focus on somatic expansion, which is that perpetual expansion of the CG repeat that causes HD. And he's specifically interested in a protein called PMS1. Chiara carried out her postdoctoral training with Dr. Frederick Sadu, investigating communication between brain cells using microscopic chips that mimic circuitry in the brain. She recently accepted an independent position and is focused on defining molecular mechanisms that underlie the breakdown of brain cell communication in neurodegenerative diseases like Huntington's. Thank you all so much for joining us today to share your work. Before we start, I want to remind our audience that you can ask questions in the Q&A box throughout the talk. So this is the Q&A box, not the chat box. We're going to have time at the end for a Q&A with our speakers, so please send your questions in as you think of them. We'll try to get to as many as we can. And also, this talk will be recorded, and it's going to be available in about 2 weeks, both on our website and on our YouTube channel. So if you want to watch it again, or if you want to share it with someone that you think could benefit from it, it will be available for that. Thank you so much, everyone, for being here again, and I will pass it over to Katherine.
All right. Thank you, Sarah. Hi, everyone. My name is Katherine Croce, and I am a postdoctoral fellow at Columbia in the lab of Dr. Yamamoto. Columbia University Medical Center is in New York City, all the way uptown in Washington Heights, and I have been conducting research here for over the last decade. And although my location has changed minimally, my interests have evolved over the years. I've always been fascinated by the brain. Initially, my work focused on brain circuitry and function, but over time, I was becoming more curious about the dysfunction of the brain. And this led me to apply to the pathology PhD program at Columbia to study neurodegenerative diseases. During this time, back in 2017, I was introduced to the Hereditary Disease Foundation. I had the opportunity to attend an HDF workshop as a scribe, rapidly taking notes on the discussions between top scientists advancing Huntington's disease research. At this workshop, I witnessed something I had never seen before, where scientists were not competing with one another, but instead collaborating on a common goal, which is to find a cure for HD. This workshop was a defining moment in my research career that ultimately motivated me to study Huntington's disease. Moving forward, my PhD work focused on a protein that I'll be discussing today called alpha. And after I finished my PhD, I made a discovery that could take my work to the next level therapeutically. And with this finding, I decided to continue my work on alpha and Huntington's disease as a postdoctoral fellow. And my hope and goal is one day bringing this research from the lab to those with HD. And today I'm excited to share some of my most recent work on this project, which is removing protein aggregates to slow Huntington's disease. So today's talk focuses on a pathology called protein aggregates, which are found across many neurodegenerative diseases, including Huntington's. So in the genome, there is the Huntington gene, which has few CAG repeats and ultimately becomes a Huntington protein. But the genetic cause of Huntington's disease is due to an expansion in the Huntington gene, which causes an atypical protein with longer than normal repeats, as shown here in brown. These expanded repeats cause the atypical protein to build up and clump together, forming what we refer to as protein aggregates. And under the microscope, we can look at this on the cellular level, and these clumps of protein aggregates look like the brown speckles and dots shown here. And I'm interested in further understanding if these clumps of Huntington protein aggregates are contributing to HD and causing the disease to get worse. For example, if we're able to remove these aggregates from the brain, will this slow HD progression? To begin addressing this, we need to know how we can remove these aggregates from the cell. And the cell has a pathway to do this, and it's similar to the game called Pac-Man. If you're unfamiliar, Pac-Man is this yellow character that's very hungry, and it goes around the game eating as many of these pellets as it can. Well, the cell has its own version of the game, and it's called autophagy. In the cell, Pac-Man is called an autophagosome, and it goes around and eats or degrades any debris that the cell needs to get rid of. In HD, some of this debris includes the protein aggregates that have been building up over time. And you can imagine that there's a lot of debris for the Pac-Man to eat. So how can we prioritize getting rid of the protein aggregates? Well, lucky for us, there is a signal specifically for the Pac-Man to target protein aggregates, and this is known as alpha. Fortunately, this is a protein we all have, and it's highly expressed in the brain. Alpha's job is to bind specifically to the clumps of protein aggregates and signal to the Pac-Man to remove them from the cell. So given its function, what would happen if we had more alpha? Could we make the brain better? Ultimately, we wanted to know if increasing alpha in HD mice could remove aggregates, and if so, will this slow the progression of HD? We're able to study HD in mice by replicating both the symptoms and pathology seen in humans. There are many symptoms associated with HD, including changes in motor function. And in HD mice, we're able to test these motor changes by having the mice perform different behavior tasks, which I'll describe later on. And as I mentioned earlier, when we look under a microscope, we could see this pathology of Huntington protein aggregates shown in brown. And similar to humans, these HD mice also have these aggregates. Another change that we see in the brain is inflammation, where these dark brown cells become large and activated. We can also see inflammation in the HD mice. So taken together, by using HD mice, we can study the motor symptoms and the different brain pathology seen in humans. The HD mouse model I'll be showing you today is known as the N17182Q. These mice have a fast progression of HD, which allows us to study changes in behavior and the brain that don't occur in humans until late adulthood. However, for these mice, it's only a matter of weeks. N171 mice begin getting sick and showing symptoms by 10 weeks old and have a lifespan of typically 20 weeks. So although it's a very compressed timeline, it allows us to gain information quickly. So as a reminder, I wanted to know if increasing alpha will remove more protein aggregates, and ultimately, if removing the aggregates will slow the disease. To do this, we used a delivery approach called an AAV, which you may have heard about from clinical trials. Put simply, it's a therapeutic way to deliver cargo into cells, and our strategy is to use this delivery approach to target and increase alpha in the brain. So once the mice got sick and showed symptoms, we tested them for a motor task and used this as a pre-treatment time point. Then, mice either received an injection of saline or the AAV to increase alpha. They were aged to 19 weeks, where they were tested again for a post-treatment motor task. And following this, we looked at their brains. At 19 weeks, the HD mice that received saline displayed protein aggregates in the brain as expected, and these are indicated by the red arrows. But we were most interested in seeing if increasing alpha could reduce the number of aggregates. Excitingly, HD mice with increased alpha had significantly fewer aggregates. Therefore, the answer to our first question was yes, that higher alpha led to a reduction of protein aggregates. Next, we were interested in understanding if by removing the aggregates, could this have an effect on other HD pathology, like inflammation? In HD mice that received saline, you could see brown staining of cells that indicates an activated inflammatory response in the brain. However, mice with higher alpha were able to reduce this inflammation. So taken together, we were able to show that by increasing alpha and removing the protein aggregates, we were also able to reduce inflammation and slow the disease in the brain. So while these are exciting results, just because we're improving the changes in the brain does not necessarily mean that we're improving the symptoms of disease. So we were most interested in seeing if clearing the aggregates improved the motor changes of the HD mice. The behavior task we used to test these mice is called the accelerating rotarod. But to explain this task, it's actually very similar to something called log rolling. Mice are put on an axle like the log shown here, and the axle begins rotating slowly and accelerates over time. Control mice, which are those that do not have HD, can stay on the axle for the entire session, which is roughly 300 seconds, as shown here in green. However, if a mouse exhibits issues with motor function, they fall off the axle as it accelerates, and they land on the platform below. We found that HD mice are already symptomatic by 10 weeks and cannot stay on the rotarod. And you could see that the data point for HD mice is lower than the controls. When retested at 19 weeks, the HD mice that received saline became even more symptomatic and performed much worse. And this is shown clearly by the red line decreasing from 10 to 19 weeks. So given how much worse the HD mice progressed, we wanted to know if increasing alpha and removing the aggregates could improve the symptoms and slow the disease. So before getting the treatment, these HD mice were also symptomatic and falling off earlier than the controls. And excitingly, after increasing alpha, the HD mice were able to maintain their performance, shown by the data line going straight across the 2 time points. You could see this dramatic difference at 19 weeks between the 2 groups of HD mice. Therefore, the answer to our question was yes. By increasing alpha and removing the aggregates, we were able to slow the disease progression in HD mice. So to summarize, I was interested in understanding if these protein aggregates contributed to Huntington's disease, and if removing them could slow disease progression. We took advantage of the pathway called autophagy that removes these aggregates, where a Pac-Man-like structure eats up the debris in the cell and knows to prioritize removing the protein aggregates due to a specific signal called alpha. So we asked if increasing alpha could remove more aggregates and if that would slow Huntington's disease in mice. By giving the HD mice either saline or an AAV to increase alpha, we found that higher alpha led to fewer aggregates, and in turn, fewer aggregates slowed the disease progression of inflammation in the brain, as well as motor function. So taken together, this showed that the protein aggregates do contribute to disease, and that increasing alpha was protective in HD mice. So a major future direction is how to take this information to the next step therapeutically. I've shown that this approach has been promising in mice thus far, and Dr. Yamamoto and I are actively working on ways to move this forward. As I mentioned earlier, a goal of mine would be bringing this from the lab to those with HD. Another exciting question is if alpha would be an effective therapy for other neurodegenerative diseases. So protein aggregates are a common pathology seen across neurodegenerative diseases, but what's different are the types of proteins that ultimately clump together and aggregate. Diseases that share this feature with HD are Alzheimer's disease, Parkinson's disease, ALS, and many more. There was not enough time today to discuss, but my work today actually shows that ALFI's ability to remove protein aggregates is not limited to the Huntington aggregates we see in HD. But I found that ALFI can be protective across neurodegenerative diseases, clearing the toxic proteins in Alzheimer's disease and Parkinson's disease mice. So another future direction would be to see if this work can be clinically effective for other brain disorders. And with that, I would like to thank the members of Dr. Yamamoto's lab, specifically Dr. Yamamoto, who's been incredibly supportive of my work, as well as the Hereditary Disease Foundation, with whom I received my postdoctoral fellowship and has been very supportive of the ALFI research throughout my PhD as well. Thank you.
Thank you very much to the Hereditary Disease Foundation for inviting me to speak today. And letting me give an overview kind of my journey into HD research. I am Joe Hamilton. I'm a postdoctoral research fellow at University College London. And today I'll go over some of the reasons why I got into HD research, but also I'll give a little overview of a few of the projects that I've been involved in whilst I've been here at UCL, which are involved in DNA repair and how I feel like they're contributing, or I know they're contributing towards the development of future therapeutics in the HD realm. So to start off, even though my accent has diluted a little, some of you may notice that I'm not actually English, although I live in London. I'm actually from a small town in Scotland called Dunfermline. Our most notable fact is that this abbey pictured here is where Robert the Bruce is buried, who is King of Scotland who got independence about 700 years ago. I am the first, as far as we're aware, I'm the first scientist in the family. I come from a line of miners and machinists. My dad works as a health and safety officer for construction sites. And the most physical exertion I have is pipetting every day. So we're not exactly sure where the scientific streak comes from. But my mum likes to say that it's from when I was a wee bairn or a little baby. I used to grab all the bathroom products, pour them in the bath and say, I'm just doing an experiment, mum. And it's looking back, you can see there was kind of no other trajectory for me. My undergraduate degree, I traveled a little north for it. So I studied forensic psychobiology, which is a bit of a mouthful, at a very small university in Scotland called Abertay. This feels like a lifetime ago, and it kind of was. During this time, I worked on murder investigations. I worked on, or I studied profiling of serial killers, and I sifted through hundreds of missing persons reports as part of my project. So it's slightly different from my day-to-day now. But this is kind of where one of the first turning points happened for me that kind of guided my career into Huntington's disease. So during my time in my undergrad, in quick succession, I lost 3 of my 4 grandparents, 2 of which had dementia. And it was one in particular who was my granddad. It was my dad's dad that really stuck with me. And essentially, my parents didn't want my granddad to move out of the home that he'd been in for all of his life, where he'd shared with his wife of 5 decades who had just passed. So logistically, it was taxing. And I'm sure as a teenager, looking at my parents as caregivers of his dad, I wouldn't have been privy to the extent of that caregiving role and how much it affected them. But I remember in particular towards the end of my granddad's life, like many with dementia, he had a fall at home, ended up in a hospital a few weeks before he passed. And I was allowed to go see him. And I just remember being so shocked that he had no idea who my dad was, no idea who his own son was. And I just couldn't understand that or how that would have made my dad feel. And at certain points, he started calling me James. My granddad started calling me James, which is my dad's name, because we looked similar when we were young. So from that point on, I was incredibly invested in understanding the brain and understanding and studying neurodegeneration in particular because I could see how it affected people. I started volunteering for the Scottish Dementia Working Group. So this is kind of a patient-led subsection of Alzheimer's Scotland. And they were incredible. I worked closely with the vice chair who lived with Alzheimer's. He was a previous professor of anthropology at St Andrews, and it was such a rewarding experience. And I knew that I wanted to study the biology of neurodegeneration and move away from psychology, which is most of what I was doing at that point. My postgraduate degree was at University College London, where I've been ever since, so it's almost been a decade. My master's project was focused on Alzheimer's and progressive supranuclear palsy, which collectively they fall under the umbrella of diseases referred to as tauopathies. And this is when the second turning point came. So I was already in neurodegenerative field almost, but I wasn't in Huntington's yet. The second turning point was I attended a seminar just before exam season, and it was led by Professor Chiara, who is my current boss. And this was in 2017, and it was right when in the HD field we had this big genetic study, which I will go on to mention, and it outlined what we needed to be studying in HD to get some disease-modifying therapeutics in the future, and that was DNA repair. The genetics of the disease appealed to me because it was a monogenic disease, but there was something that Chiara said that has stuck with me ever since, and I know it's stuck with other people as well, this phrase, and it was Huntington's disease is the most curable, incurable disease. And as a researcher wanting to make an impact in the realm of neurodegeneration, that is an incredibly attractive concept for obvious reasons. So the rest is history. I've essentially been working in the lab ever since for almost a decade. So I've went through research assistant position. I did a 3-year PhD, and thanks to the HDF, I'm working as a postdoctoral fellow. UCL itself is a great place to study HD because we have our own designated Huntington's disease center. So we span both molecular labs, so cell and mouse, and this includes the lab of Jill Bates, who pioneered the importance of that short, alternatively spliced Huntington exon 1 fragment in the disease. But this also includes Sarah's lab, which is where I fit in the landscape. And we mainly work with human cell models, stem cell models, and sometimes human tissue as well. But the center is also integrated with the HD clinic, and that gives us patient access that enables us to do natural history studies, to monitor patients throughout their lifetime and understand the disease in more depth. And through collaboration with HD gene carriers, we're able to look for things like in vivo biomarkers, such as neurofilament light you may have heard of. And these are essential for clinical trials going forward. So it's really a great place to work on Huntington's disease. But what do I do specifically? Well, we know from studying the HD gene that the biggest determinant of when you get the disease is the length of that CG repeat in the Huntington gene. More repeats equals a more toxic protein that can't do its normal function, but also gains functions that we don't want it to do. But even when we control for the length of that CEG repeat, we still get huge variation in terms of when someone actually starts showing motor symptoms, which is clinically noted as disease onset. Someone, for instance, in this bar, someone with 42 CEG repeats could start showing motor symptoms at 18, another at 70. So what exactly is causing that variation? I mentioned earlier that one of the things that drew me to the field was a genetic study, and that genetic study answers this question. So the data was presented in one of these plots, which I appreciate looks overwhelming, and that's kind of the point I'm making here. So this is called a Manhattan plot, as it usually resembles a city skyline. This one, for instance, is from Parkinson's disease, I'll preface with. This isn't Huntington's. But this is how they normally look. Researchers take these mutations represented by each dot, and they try and figure out which genes are these mutations associated with, which gene is nearby, what could we figure out is also going wrong in the disease, and what could be potential therapeutic targets, what attractive therapeutic targets. In this instance, the Parkinson's genes span dozens of cellular pathways. Some are exclusively expressed in certain cell types. Now, this was the Huntington's disease genetic study, which obviously looks less like New York and more like the suburbs. And even more encouragingly, everything that I've labeled here in red, they are known to interact, these genes, once they make the proteins are known to interact, and they're all within DNA repair. So to put that into context, there's tens of thousands of genes that could have come out of these genetic studies. but these few did, and they all converge on this pathway. My job has been picking some of these genes that I'm interested in and seeing how they might affect the disease and how they're affecting age of onset of HD as well. Now, one aspect of HD we're particularly interested in, as Sarah previously mentioned, is this process called somatic expansion. So that's when that CG repeat in the Huntington gene gets progressively bigger and bigger as someone ages. And we know that a faster rate of expansion is bad because we believe you reach a toxic threshold sooner. And that is when we believe neurodegeneration begins and occurs because the protein is so big and so dysfunctional. During my PhD, I started working on FAN1, which is on the right here, which was by far the biggest hit. For instance, mutations within FAN1 were suggested to modify the age of onset by 5 years. And that's just an illustration of the CG repeat gets longer and longer, and it can reach hundreds and hundreds by the end point of the disease. So I'll show you a little bit of preclinical data now. So this is from my PhD. This is one of the main cell models we worked with when we were studying FAN1. It's a cancer cell line. And what we do is we introduce the mutant Huntington gene. This one has 118 CG repeats, so it's really big. And thereafter, we monitor as the CG repeat expands over the lifetime of the cell. We're also able to explore other biology that's interesting, like protein-protein interactions, as well as the DNA repair mechanisms themselves. But what makes this cell line very special is that it is lacking normal FAN1. It's knocked out by CRISPR gene editing. And therefore, we can replace it with FAN1 variants that we're interested in that might have small deletions or small mutation. And that'll enable us to know which parts of the gene and which parts of the protein are involved in somatic expansion. So we published this work towards the end of my PhD, and it was exciting because we found 2 potential targets for future therapeutics. We first of all found that if you knock out FAN1 and don't have it, you rapidly accelerate somatic expansion. So it's playing a protective role in this context. But more importantly, we showed 2 functions of FAN1 were responsible for that protective effect. So if you made single mutations, which either prevented FAN1 from cutting DNA, we make this mutation at the end on the right-hand side of the gene here. If you make that mutation, or you make a mutation in this yellow part, which prevents binding of FAN1 to this other protein called MLH1, which came out in the genetic study as well, you cause the same effect as knocking out the gene altogether, in that you accelerate the rate of somatic expansion. This work is moving forward in our lab, as well as others, into models such as human neurons, so more relevant to physiology, and mice, so we're getting closer to a potentially druggable target. We would be looking at either modifying that FAN1-MLH1 interaction or upregulating FAN1's normal function as a nuclease, so cutting DNA. But we really need to make sure the safety of that as well across multiple models is investigated. More recently, and thanks to the HDF who fund my work, I've started looking at another one of those DNA repair modifiers that came out of the genetic study called PMS1. So what made me want to study the gene and the protein is that it's a complete enigma. We know nothing about it. It physically associates with all those other DNA repair proteins, but if you remove it from the cell, it does not affect the function of that pathway. So currently I'm in the process of doing a similar structure function study, as I just showed with FAN1, introducing variants into a knockout background. But I think today I decided to show some recent data that I think is more pertinent to this audience. So part of my work is a collaboration with Regenerative Therapeutics, and they've provided me with a tool compound designed to try and lower PMS1 protein levels in cells. So what I did is I took this compound, added it to stem cells with 180 CG repeats, so huge. And as you can see, you see a lovely target engagement. It lowers the PMS1 protein. But importantly, we see a concordant decrease in the rate of somatic expansion, suggesting that PMS1 lowering could also be another target for HD therapeutics. So the overall message is that it's a really exciting time in HD to work in DNA repair specifically, because over the last decade, we've made strides due in no small part to collaboration with HD gene carriers, but also developments in techniques such as CRISPR And I truly feel we're moving towards clinical trials with some of these candidates. I put this in here. This is actually from my PhD thesis, so I'm glad it's seeing the light of day. But this is a list up to 2022 of all the published work on these genes, and just to really back up the functional role of these proteins in somatic expansion. What you might also notice from this is that these genes are not just implicated in Huntington's disease. The same mechanisms present in other repeat expansion disorders. This includes spinocerebellar ataxias, myotonic dystrophy, as well as Fragile X, meaning there's a big opportunity in the therapeutic landscape. At present, there are a few companies interested in targeting DNA repair, namely looking at a gene called MSH3, which is another gene we also work on, and it's undoubtedly driving somatic expansion. What's also encouraging is that several of these appear tolerant to loss of function, in particular MSH3 and PMS1, which means that removing them from the patient population may be tolerated and may be less associated with cancer risk than you may anticipate. We're getting new data on these mechanisms every week now that's emphasizing the importance of somatic expansion in the disease process. Some groups are reporting that if you stop expansion early enough in mouse models, you can prevent accumulation of mutant Huntington into clumps, and the rate of somatic expansion itself may be directly associated with clinical outcomes in patients. So putting this all together, it points for DNA repair at least to an early intervention strategy where we can target the disease early in the process or early in the lifetime of a gene carrier prior to motor symptom onset and prior to severe neuronal health decline. And according to the current model of HD, if we can halt this process of somatic expansion early enough, individuals won't reach that toxic CG repeat length threshold, whereafter patients would likely benefit from Huntington lowering therapeutics more so to prevent further decline. So I'd like just to finish up by thanking HDF for asking me to speak today and helping me to stay in HD research. I'd also like to thank my lab. All of our work is hugely collaborative, and this doesn't even encompass people who have worked on these projects and have left the lab since, and has similarly helped us progress. So thanks to everyone, and thanks to everyone for listening as well. I'll now hand over to the next.
Hello, everyone. I'm Chiara Scaramuzzino, permanent researcher at the Grenoble Institute of Neuroscience in France, in the team of Frédéric Sodou, a team that is on the intracellular dynamics and neurodegeneration. First of all, let me thank HDF for inviting me. I'm really honored to be here today to give you an overview of my project, whose main aim is to target and restore transport of survival signal to cure Huntington disease. I did my PhD studies in the beautiful city of Genoa, where I developed a strong interest on the molecular mechanism underlying the neurodegenerative processes occurring in neuromuscular disease. I then moved for my postdoc on the French Alps in the lab of Frédéric Sodou, where I started to focus on HD. In the last years, I had the precious opportunity to get in touch with HDF that is supporting my research, which has the ultimate goal of unrevealing novel defective mechanisms that are occurring within the brain during HD pathogenesis. As you might notice, all the work I have done has been about the brain and the pathology of the brain. Now, our brain is a network made up of a multitude of neurons that are interconnected and communicate between them. Within this network, the information are moving from a given neuron A to a neuron B. The information and signals that are transported are critical for the correct maintenance of the neuronal network formation, such as the one connecting two brain regions as the cortex and the striatum. Inside the neuronal network, the information are transported by small organelles that can move in two ways: anterogradely from the body of the neurons toward its periphery, and retrogradely, which is the opposite direction. Within this region, the transport of the two specific proteins is critical for the integrity of this corticostriatal network, such as the neurotrophin BDNF and its receptor TrkB. These signals are transported by several organelles that we can visualize as vehicles moving within streets in two opposite directions, like for example car or trucks. For this reason, I like to imagine that the neuronal network in our brain is like a roadmap. For example, in a roadway, there are millions of vehicles that transport information from one point to another one. And more than car and trucks, we need also the transport by ambulances of critical supplies to hospitals. Here, for example, we need to deliver urgent supply from the University Hospital in the south of Grenoble to the Nord Hospital, similar to the retrograde transport of TrkB receptor within neurons. What I recently discovered is that on some transported organelles, it exists a kind of a navigator, a GPS, which is composed of two proteins, Huntington and calcineurin. We discovered that this protein complex is capable of ensuring the correct direction of retrograde transport of urgent supplies, such as, for example, the one transported by TrkB, from the south to the Nord Hospital when necessary. We also discovered that on the same organelles carrying this TrkB receptor, it exists also another protein, the creatine kinase B, which is responsible of providing the fuel necessary to initiate this retrograde transport from the south to the Nord Hospital when necessary. Having understood this mechanism, what I'd like to elucidate are the different signals that are carried by these TrkB containing organelles and what they are used for. For example, they are transporting drugs or blood samples by ambulances. To do this, we reproduce this corticostriatal network on a microchips, and we specifically studied the dynamics of what we call this TrkB retrograde transport from striatum to the cortex, as, for example, it occurs by ambulances from the south to the Nord Hospital. So the communication between cortex and striatal areas takes place with this neuronal transport mechanism that in healthy individual, this information is transported in both directions, this ensuring the supplies everywhere within the brain. When neurons begin to degenerate, there is a reduction in connection between the different brain areas, which is also due to a reduction in transport within these two areas. As for example, there is the transport of less urgent supplies between the south and the Nord hospitals. This is the case of Huntington disease, where the transport is disrupted by a mutation affecting the Huntington protein, leading to X dysfunction. Indeed, what we know is that the transport in Huntington disease is altered, and what we recently discovered is that this TrkB retrograde transport is defective. And we also have evidence that this is due to the fact that the GPS does not work properly, and that the transported survival signals are reduced. We also obtained data that report that the fuel is also missing as TrkB, as the CKB dynamics are altered. So we are going to investigate in details this mechanism. And also what we are going to investigate is if there is a reduced or modified content of the transported supplies. So to conclude, since in healthy brain, this is how it works, where Huntington is involved in the traffic of vesicles transporting signals within the corticostriatal network, this project will enable us to better understand, in particular, the function of Huntington protein in transport of TrkB receptor from the periphery of the neurons towards the body of the cortical neurons, and to investigate the positive outcome. But also this project will elucidate the impact of the mutation occurring on HTT that we have in HD, where this transport is dramatically impaired, thus causing a reduced transport of fundamental signals and supplies in both directions. Moreover, we already demonstrated that improving the transport of BDNF vehicles within the cortical striatal network, in particular from the body of the neurons toward its end, as from the north to the south occipital, is ameliorating HD phenotype. And what I propose here is to rescue also this retrograde transport of track B ambulances in order to increase the survival signal transported in the cortex. Moreover, I will potentially identify novel therapeutical targets for HD that at long term might be also relevant for other neurodegenerative disorders involving these transport defects. I would like to thank you, all the members of the lab, the funding agency, but in particular HDF, for including me in this fantastic folk community and for supporting this project, my career, and my willing to contribute finding new therapies for HD. Thank you all.
Awesome. Thank you all so much. I love all of the analogies that you guys used. I think analogies make things super easy to understand. So I love the trafficking up and down is a really great way to understand transport. And then Katherine, your Pac-Man was awesome. Okay, so turning to the questions, please feel free to keep submitting questions throughout the Q&A. We'll get to as many of these as we can. And they can be technical, non-technical, just any question you might have. All right, so we'll start with one for Katherine. So the person says, looking at the ALFI results that are very encouraging, do you see ALFI moving into the clinic soon for human application? Does the ALFI concept work for different transgenic mouse models of HD?
Yes, so great question. I don't know about soon, but our goal is to bring this to trials as soon as possible. And yes, I have replicated my findings across mouse models of HD in both transgenic mice as well as knock-in mice. And I found consistent results that increasing ALFI is therapeutically relevant.
Very cool. Maybe I'll add to that. Have you tried it in like human-derived stem cell models or have you tried it in like human-derived models at all?
I have not tried it in human models, more so just proof of concept of, are we able to increase ALFI in cells as well? And we are. But no, I haven't done any additional work on that.
Okay. And this question kind of piggybacks on that about increasing ALFI in cells. And I might throw this to you first, Joe, and then back to you, Katherine, since you both have a target that you're looking to overexpress. So the person asks, how do you see increasing these targets? Would people take a pill or is there another way to boost the signal? So maybe Joe, you could go first and then Katherine.
Yeah, some of them, it depends on the mechanism of action. So if we go similar to the Huntington lowering ASO, that could be an intrathecal lumbar puncture if it's something long-acting to upregulate FAN1, for instance. So it's an antisense oligonucleotide, you got a lumbar puncture maybe once every 6 months, something like that. Something to bear in mind with these genes in my instance for DNA repair enzymes is they seem quite tolerated, specifically in the brain, the ones that we want to target. However, they do have associations outside of the brain with cancer risk. For instance, the other DNA repair enzymes that we're less keen on for therapeutics are more associated with things like colorectal cancer. So if you would have something like an orally delivered therapeutic that would reach the peripheral system and therefore increase cancer risk potentially with those. So there's kind of like pros and cons either way with potency versus off targets. So it's up to the companies to decide. We would need to see the safety profile first, to be honest. We would need to see some effects in mice and see some long-term biomarker monitoring as well.
In the same vein, in the same vein as what Joe had just said, one way that we could increase the proteins such as ALFI would be through a type of AAV approach that's been used in humans so far. And this would have, similar to what Joe just said, kind of longer acting effects, maybe every 6 months or so, but those could be a little more invasive. So we're also interested in currently exploring other systemic approaches, whether it be intravenous. A pill would be ideal. I don't know about having the right partnerships or collaborations for that at the moment, but that would be exciting as well.
So I'll throw one to you, Chiara, since we're kind of talking about drugs that could target some of these things. Are there any drugs that already target neuronal transport that maybe you could take advantage of, or would you have to completely develop a new drug for what you're finding?
I think that for the mechanism that we are really looking specifically, we need to really like develop something that is going to picky either the interaction between the proteins, either specifically the endosome transport. There are drugs that you can use to improve transport, to accelerate transport, but to really look at this specific mechanism, we really need to have like what we are developing, for example, in this moment is to study the interaction between the two protein composing the GPS, so Huntington and calcineurin, and to see if we can try to force this interaction in order to promote the transport of tract ambulances towards the neuronal body.
Okay. So I think this one could go to any and all of you. So maybe you could just answer in turn. So it says, it was great to hear all of these different approaches to looking at potential HD therapeutics. Can any of you comment on the importance within the research field to focus on multiple targets, especially since there are so many different issues happening with HD, as well as how this might all work in the future? So what do we think about combinatorial therapeutics?
I think that this is really like the best approach is really to look at different mechanisms, as Huntington has several functions within the neuronal cell, but not only neuronal cells. So I think that combining multiple targets and having multiple approaches and focuses also, it's the best way to really like try to conclude and arrive to something that is really helping the patients.
Katherine, Joe, do you guys want to add anything about combination therapies?
Yeah, I think we've learned our lesson about put everything in the one basket. I think we definitely need to have multiple approaches, and I would say they all have their advantages as well. And in my mind, there's three big disease-modifying things to target, which would be somatic expansion, the Huntington protein, as well as exon 1 of the Huntington protein. And I think, just to caveat my own kind of therapeutics, potential therapeutics, these, for the most part, what we currently understand is these would prevent expansion to a larger CG repeat size, which is going to be more toxic. However, there's always going to be individuals who you're not going to be able to get before it gets there. And at that point, Huntington lowering would probably be much more efficacious at that stage. It would help prevent further neurodegeneration. I would say in the case of MSH3, there's evidence that it could even reverse expansion to a more normal range of seizure repeat size, but that is tenuous evidence so far. So I would say that we couldn't just invest everything in somatic expansion. You're going to go for Huntington lowering, and we know how toxic the exon 1 product alone is as well, and it can change your entire the biology of your cell, the transcriptome, and causing of aggregates as well. So it could help with that. But I definitely think multiple approaches you need.
So maybe since you mentioned it, Joe, maybe you could explain a little bit about what is the exon 1? What is this fragment you're talking about for our non-scientists in the audience?
Yeah. So exon 1, essentially the part of the gene where the CG repeat is. You can either produce the entirety of the gene or just that small part with the CG repeat if the CG repeat is in the disease threshold range. And we know that even mice just expressing that small part of the gene with a disease range CG repeat is toxic. It can cause aggregation, it causes motor deficits. So we know that alone is toxic. And there's recent work from several labs showing that if you lower in mice expressing the whole toxic protein or just that short bit, if you just target that short bit, it's hugely therapeutically, it has huge therapeutic potential. It corrects a lot of what we call phenotypes, but behaviors, that kind of thing. Things that go wrong in the cell, you can revert or prevent just by targeting that. So we know that's really important in the disease mechanism.
Awesome, thanks. So Katherine, I want to ask you, because I actually don't know this story, and so I'm interested, but how did you guys identify Alfie? I know Joe talked about these genetic studies, and this is where a lot of people identify these modifiers, but Alfie is not part of that. Am I correct? So how did you guys figure
So
out
Alfie was separately, it was actually identified in a GWAS looking at HD modifiers, and we found that a SNP in ALFI was actually protective. It delayed age of onset in HD patients by 10 to 22 years. So it's very good. But ultimately, ALFI was discovered by one of our collaborators, Dr. Ann Simonsen, and she is in Oslo. And she and my PI, Dr. Ai Yamamoto, have been working on ALFI since then, roughly 2009. And they ultimately look at ALFI in development and in cells. And while we're looking at it in the context of Huntington's disease.
Awesome, thanks. So Chiara, here's one for you. They say, great presentation. Can you comment on why the transport of BDNF is so important and what it is about the TRK beta receptor that does and why it is important?
So BDNF is the neurotrophin that is responsible of maintaining this cortical striatal circuit that I talk about, so the one that is degenerating in Huntington disease. And actually, the neurotrophin BDNF is transported within cortical neurons and is released in the synaptic at the synaptic cleft, where it binds the receptor TRKB, which is on the striatal neurons. So then the TRKB receptor is taking this the neurotrophin and is internalized and transport the signal of survival in striatal neurons. So it's really important to maintain this connection. And actually what we know is that striatal neurons, they do not produce BDNF. So actually cortical neurons are the main source of the neurotrophin for striatal neurons. So for this reason, the neurotrophin and the transport of this neurotrophin is really important to maintain the integrity of this neuronal circuit. And TRKB is really important because it's the one that is receiving this neurotrophin and is transmitting the signal inside the cells. So, and it's either important for both striatal and cortical neurons.
Thanks, Chiara. So here's one for Katherine and Joe. They say you both talked about different approaches, reducing the Huntington aggregates and working to prevent expansion of the CAG repeat. Do you think that the mutant Huntington protein clumps occur when the CAG repeat has reached that threshold you mentioned, or does it occur with the normal repeat size?
So these mutant Huntington protein aggregates have been correlated with CAG repeat size. But just something to note about protein aggregates in general, not the Huntington protein aggregates, is these can also form just from a misfolded or something going wrong with a protein, and it ultimately clumps and becomes one of these aggregates, and so through autophagy it can be cleared. So this is a faulted process that does happen in those without a neurodegenerative disease. But yes, specifically for HD, there has been correlation between the two.
Yeah, I would add on to that there's been a correlation. They've shown that essentially the longer the CAG repeat, it's more opportunity for production of exon 1 that I previously mentioned, and that is such a good seeder of aggregates as well. So it could be a mix of the two as well, yeah.
All right, thanks, you guys. All right, so we are at the end of our time, so I'm going to wrap it up here. But I want to end by, again, thanking our sponsor for this webinar, which is Neurocrine Biosciences, as well as everyone who donates to the HDF to support the research that we fund, our audience, and last, but of course not least, our speakers. Thank you all so much for giving your time to be here today. This is the last webinar of our 2024 series, but we are working on an exciting lineup for 2025. We're going to kick things off in January with a webinar where I will recap research highlights from 2024. I will cover the monumental year that we've had in clinical trials so far, as well as other exciting up and coming areas of research. So you definitely don't want to miss it. Stay tuned for details about that. Thank you all so much for joining us today, and we hope to see you at our next webinar in 2025. Thanks, you guys.